B, new variability. New variability. Okay. So there are two different types of chain for the recognition of antigen molecules. One is the large combining site with greater affinity. Another one is the new variability. Suppose you get one for this large combining site, but you also have the same combining site. You should have various recognition of antigen variability. So once this particular antigen 1 can recognize here, but suppose if antigen 2 which is coming up, it cannot bind here, so you need to have the another part of antigen variability. So this variability of to bind with the different antigen, that is also being done by interchain amplification. So that is the message here. So interchain amplification will help to bind with antigen molecule by large combining site with greater affinity, number one. Number two, the variability of different type of antigen to bind, the capacity to bind with different variety of antigen, that is also possible using interchain amplification. So what are those chains? We are talking about this, right? What are those chains?
Dr. Sama?
Yes.
Can you please use the marker? Because when you use a pen, we can't read it when you write over there on that
Oh, on this one?
display over there.
Okay.
Yes,
let me
Thank
go
you.
over there. Let me check the marker. What are those chains? Can you see them? Yes.
That is so much better. Thank
Oh,
you
okay,
so
okay.
much.
What are those chains? Okay. So you know about the antibody molecules like a heavy chain and light chain. Okay. And they have a pairing mechanism. They are always paired. Heavy chain and light chain paired. For example, in this antibody molecule. Okay. So in this, what happens here? Heavy chain is made up of, is a two B cells. Two B cells are responsible for making the two heavy chains. Okay. But at the same time, the same two B cells, which will have a different light chain. Okay. So, for example, you have a B cell and B cell, and they will produce same heavy chain, but the same B cell will have is a different light chain 1, and this is a different light chain 2, different one. And this light chain will have a different specificity, and this is a different specificity of binding. So in my earlier discussion, I'll just to show this, the larger one which is coming from of a different B cell, but the same type of B cell will have the variability, and this is coming from a light chain, and that is a specific for a particular B cell. I'll draw in a different way to explain this mechanism. This is a B cell and this is another B cell. Okay, and they both will have the same heavy chain, H for heavy. Okay, but it will synthesize a light chain 1 and light chain 2. They are different types of light. This is the same one. H are the same. But what will happen when you combine? These are the B cell. Okay, B cell. When they combine HL1, here you have HL2. So the heavy chain is the same. The arm, very big one to bind with the antigen molecule, is the same one, heavy chain. But here, this is the light chain, will have a different type of antigen binding capacity, and here the L2 will have a different antigen binding capacity. So in that way, this gene is rearranging in a way, or interchain amplification in such a way, it can bind with a different antigen At the same time, you have a heavy chain is the constant heavy chain. Do you follow that concept? That's great. Now we will go on to the major diversity and specificity which occurs on to these light chain, how they will do it. Okay. Major diversity and specificity okay, and they are coming from heavy chain, comes from heavy chain. So I was discussing earlier the specificity as well as the diversity, and they are coming from major from the heavy chain. Right, in the example is CDR3. Okay, and this results of this variability, what will happen? 230 functional T cell receptor, 230 functional, and then 160 same thing functional germline segments. Okay, so the calculation for the diversity and specificity, it comes from the heavy chain, but the arrangement, the example here is the CDR3. This is our type of receptor, name of the receptor. Okay, don't panic the name. This is only a name of the receptor, CDR3. And it not only this, but this diversity will give rise to 230 functional T cell receptors and 160 functional germline segments. Okay, and these different combinations of this and this, I will go on to the next slide. 230, right? And that receptors will give you that gene, and then that give rise to 4.5 million. Okay, and this is for 160 germline segment. This will 2.4 million. This one 2.4 million. And these are the different combinations. See, 230, and that for the different combinations of these particular segments of this particular sites are the functional receptor. 230 becomes 4.5 million receptors it can produce, that particular arrangement. The same thing, 160 segments, gene segments which is present, it can give rise to 2.4 million receptors at different molecules of the protein. Imagine, how do they do it? This is one of the excellent way of explaining the evolution of the organism, how they can pair, how they can rearrange, how they can induce so many number of receptors in our system, and that can easily recognize this. In other words, if we evolve from one single organisms and the human beings can recognize this number of species of different type of protein by our own B cells, we have the capacity. If we are not evolved from this, what will happen? We cannot recognize anything on that. It's its own and it's different. So we have our ancestry of the evolutionary pattern, and these are the proteins which is more significant, which can bind with this one. So that's what the evolutionary theory, which is going back, and they come, oh, this is excellent theory which we can correlate, without which we cannot explain the evolution. That's what one of these immunological reactions going on. Okay, so the next part is the somatic hypermutation. That's also an amplification reaction, like a somatic hypermutation. See, in all our discussion today and the last week, as I mentioned before, to find out how many ways of getting our receptors, different types of receptors, how many we can synthesize, how many we have already established. So we have only 230 genes and 160 genes, and total it's going to be around 400. Around this 400 and gene, and you are making 4.5 million and 2.4 million of receptors. So that's the question. So another way to explain this mechanism is somatic hypermutation, and this is on immunoglobulin Ig V region of gene, V region, immunoglobulin and V regions of genes. Okay. And it can undergo significant, the V region can undergoes significant somatic mutation, somatic hypermutation. Hypermutation. Okay. This hypermutation is more frequent in IgG. Okay, this is more in IgG, in immunoglobulin G. Okay, more frequent this hypermutation, and IgA, and then IgM. Okay, antibodies. I'll write it in a next slide. Perhaps that's maybe reason. Somatic hypermutation. Hypermutation, okay, is more in IgG and IgA than IgM antibodies. Okay. So this part you get the more variety, more number now is coming out. Okay. Then the another theory is the T cell receptor gene. Receptor genes do not undergo somatic hypermutation. Somatic hypermutation. T cell receptor, TCR genes, these genes do not undergo somatic mutations. So I'm just underlying, I'm just giving these slides because sometimes, you know, I will ask questions specifically on this. So B cell receptor gene do not undergo somatic hypermutation. True or false? It's a false, right? Because the T cell receptors, that's the only things that do not undergo. The B cell receptor, what we discussed earlier, that is the B cell. That will undergo hypermutation. So like that, I will ask questions from my lecture series. So if you don't attend the class, I'm trying to put it on to the video lectures, and at least in the exam time, you can go through it. But certainly, I'm going to ask some questions from here. Okay, that's why I ask you to, you know, get the summary of the lecture. So that's your homework. So that's where you practice for your exam as well, along with your regular multiple choice questions and short answer questions. Okay. Sometimes, yes, you will get short answer questions from these lectures series. Okay, get it. So now I shall go on to the NK receptors. NK receptors. Any questions so far on the hypermutation and receptor editing? First of all, you should know what is the objectives of this class. Just the objective of the class to understand how many variations or variability or number of receptors which is present in the B cells, and to recognize the different type of antigen. Otherwise, how do you synthesize antibody without recognition? That's why I put a simple example like these B cells are the blind cells, and these blind cells can operate by Braille method of recognition. What is the Braille method? You know about it? Touch and feel and then recognize. The same thing, the B cell will have the protein, and that protein will act as a hand and then as a Braille method to attach to the different type of protein in the bloodstream, and then recognize the one which is more suitable by method of attraction or some sort of magnetic forces or some bond formation between the antigen and these receptors, and then recognize, and thereby the virgin B cell will transform to the P cell or plasma cell, and that plasma cell produce antibody as well as they have more number of receptors. So my question is, how do we have this variability onto the receptors? And that receptors is coming from, which we discussed, interchain amplification, receptor editing, and somatic hypermutation. And we have only 400 and something, right? 230, another one is 160 different sets of gene segments. Only that we have it. But we have the foreign molecules or foreign invaders, or you can have a bacteria or virus. We have thousands and thousands and millions. How do we recognize with 410 genes to recognize all these genes? Unless otherwise, we have to have so many receptors or protein, specific protein. So to synthesize that very number, this class, what we discuss about so many variability. So you should understand this principle so that we can go on to the next step very easily. Do you follow these steps? Yes? Sugarland? Synchrorange? Do you follow why we are discussing today now? Yes? Okay, good. In Victoria? Is Kelly there? Okay, good. NK receptor, NK here we call it as a natural killer cell. Natural killer cells. And this natural killer cell licensed to kill any of the bacteria, like a James Bond here, this one, NK cells. Okay, 007 is coming out now here. And these natural killer cells will have its own gun. It's in a like a cytokine, and then it will recognize the cells which possess, I put it in a way, which possess the virus particle or the antigen or the bacteria. So it can identify. So the receptors, natural killer receptors, should now recognize the antigen. As I mentioned before, the bacteria are viral and they are the antigenic protein, and it may hide, it may evade, it may not recognize by T cell receptors, it cannot recognize by the B cell receptors, it escapes all these routes. Then finally what will happen? It will get into any of the cells, liver cell or the brain cell or the stomach or the intestine, or any cell it can invade. It can go inside and hide. So B cell will recognize those cells? No. T cell recognize? No. But the NK cell receptors, and they can sense which these cells are invading into the other cell. Suppose this is the cell and this is our host cell. I can put the L for the liver cell. And here this is a person, this is the virus particle which is getting in and can go in. And then this is a bloodstream, but it cannot be recognized by the bloodstream here. So bloodstream cannot recognize, of the other cells cannot recognize, and the B cells cannot recognize, but because it is there. It is hiding inside. So there is some specific mechanism to identify this cell, to invasion, and then to kill the entire cell. Not the normal liver cell, but only those cells which is infected. So the natural killer cell will destroy this. So to destroy this, what you need, what is your equipment? As I mentioned before, it should have like a hand, right? To like a Braille method to have these receptors, and the receptors will go and sniffing around the cells. Hey, is there anybody's invasions going on? Is anybody is hiding inside of any other cell so that I can go and destroy? So these NK cells, they have a specialized receptor, specialized protein, specialized membrane protein which will recognize the invasion. How? That's our question now. Okay. Now there is a molecule which we are going to discuss earlier, but I'm going to explain now. So the molecule is MHC class 1 molecule. Okay. We are going to discuss probably today or the next class as well. So this molecule normally in a general normal cell, it is going to be outside. This one. Most of the cell we are going to discuss later, but these are the MHC molecule and these are the protein, and these protein are present in the membrane. Okay, so now, so the NK cells or the natural killer cell can recognize this, and then okay, this is a normal cell. At the same time, if you have an infected cell, I put the liver, yeah, it's an infected cell, they don't have this MHC molecule, MHC1. It is present positive, here the positive. But here this cell is negative because this is being invaded by, or somebody is hiding inside this one. This is the I put the viral infection. So this is the normal cell will express this MHC molecule, and the NK cell will not touch, it will not kill this cell, but at the same time when the NK cell or the natural killer cell will see here and there is no MHC1 molecule, and what it will kill this particular cell. So now you have a morphology of a cell. As I mentioned before, NK cell will go roam around in the blood as well as the tissue surface, and it will see the MHC1 molecule. Okay, they are normal. Okay, they are fine, and this cell will inform outside or to NK cell or to any other cell. Here's MHC1 is synthesized, is happy. I am happy, so I am giving a broad smile here. But the infected cell, in a way, the virus will not know about this MHC1 molecule, but as soon as getting in, this cell feel uncomfortable. Hey, somebody is unnatural, is getting inside my territory, so I have to communicate to outside. How can I communicate? And this is the a language which these cells will speak. These cells, most of the cells, they have a MHC molecule as a language, as a molecule which will express outside the cell. Once the cells are outside, is they happy and then gone. But at the same time, if these cells will invaded by the virus or bacteria or anything like it escapes the B cell, it escapes the other receptor, it escapes the antibody, as well as getting into the liver cell and using these machineries or synthesizing this, and thereby it invade the another cell. If it invades, these cells will immediately stop synthesizing this MHC molecule and send to this surface area. So surface area is going to be a blunt one, and there is no MHC1 molecule. And thereby the NK cell will come and see that, hey, there is no, and here it is, here is this guy a hiding place. So it will come and destroy this particular cell. So this is a natural way of killing the abnormal cell. Do you follow that? Yes, go ahead.
Dr. Soma, so if for some reason, if the antigen get in the cell and for some reason the cell they still retain the MHC1
Right.
outside, so the
Right.
NK cell will not be able
Will
to
not
kill
be
it,
able
right?
to recognize, no.
So they only recognize of the
Right.
MHC cell outside.
Outside, yes. So that's the thing. If it gets inside, the cells will not synthesize the MHC1, and thereby it gives silent communication to the NK cell. The NK cell is roaming around, then it will see, hey, there is no MHC1, no MHC1, so go on attack, and destroy that cell. The entire cell is destroyed there. So that part, the mechanism, we will see on the next class how it will go, but we will see the next couple of things, and then we will go into the details. Okay. Do you have any other question in Victoria? Okay, in Cinco Ranch? Okay. Now, why these cells are lacking the MHC? Why the MHC1 class 1 molecule are lacking these cells? And which cells are lacking? One good example here is the malignant cell. Malignant. What is malignant? Huh? Microbe infection. Malignant cells are cancer cell. Do you know that one? Malignancy, cancer cell, right? So the cancer cells also, they don't have MHC1 molecule. And the microbe infections, as I mentioned before, that also lacking the MHC1 molecule. So NK cell can happily attack this and kill. And you can also extrapolate this NK cell also attack the malignant cell and destroy. In that way you can find a cure for cancer. Correct? Yes? So in probably 20 years back, people thought, yeah, we found cure for cancer. We will increase the number of NK cell, natural killer cell, and dump it in the bloodstream. More NK cell, or activate the NK cells in our body, and this NK cell going around and around and around and kill wherever the MHC is not there. Kill it like that. But it is partially successful at the time. But eventually the cancer is learned that technique, and then it can also start synthesizing the MHC1 molecule. That's a bit complication. But most of them, in general, malignant cell, they don't have the MHC1 molecule. Okay, that's you should understand. And microbe also, it is there, the infection. Yes, MHC1 is not there. Thereby NK cell can recognize and these both and can destroy this malignant cell. That part. So if you see this mechanism in the how the receptors of the NK cell will activate and kill, and we will see that one in just a second. Suppose this is the cell, okay. I put that one NK cell, okay, and our topic is the NK receptors, right? And these receptors is over here. There are 3 receptors are binding, okay. This one receptors, this is receptor number 1, okay, and this receptor can activating receptor, and this one inhibiting receptor. And then the another one can bind with the like a FC fragment, the FC gamma R3 receptor, and they will bind to antibody coated target cells. So this is the NK cell, the natural killer cell. And this natural killer cell will have receptor 1, receptor 2, receptor 3. So if I ask a question about write short notes on NK cells or NK receptors, so you have to say NK receptor or NK cell will kill the MHC1 molecule lacking normal cells, or the infected cell or malignant cell where they lack the MHC1 molecule, number 1. And the NK cell, they possess the 3 types of receptors. One is the activating receptors, inhibitory receptors, and third one is the antibody coated target cell attacking receptors, or the binding with the antibody. So this is number 3. So when they invade a cell which an infected one, okay, so it will bind. This is the infected one, okay. So there is lacking no MHC molecule, but it can activate, it can bind here, and then the infected cell will be injected by like a cytokine. Cytokine is a bomb, like, cytokine, a hand grenade. So NK cell will bind here, and then it will kill this infected cell by using the cytokine. It binds with the receptors, and then it will recognize, because there is no MHC1 molecule, and thereby it can kill, can induce cytokine, and then it will induce as apoptosis or cell suicide molecule, caspase A, and that will chop up all those DNA which is present in the nucleus. So that will kill eventually. This is a one type. Suppose these NK cells will bind to another cell which will carry. Suppose this is the antibody coated. The green is the antibody coated, and then inside this you have another cell, and this is the cell, microbe infected cells. Which is the cells which is infected with microbes, and mostly these are coated with antibody. This will recognize, this antibody coated recognize, and then it will bind, and then it inject the same cytokine and kills the cells using 3. So it is not with the activating receptor, but Fc receptor alone it can bind here, and thereby it can induces the cell death. Okay. This one, inhibitory receptor, is the one which will recognize the MHC molecule. It will recognize the MHC molecule. Suppose a cell, I'll draw the same inhibitory peptide like this. I'll put it in this way. So for my explanation over there. Okay. So there is a groove, and this is a normal cell, N for normal cell, and they have the MHC1 molecule here. Okay. And this molecule will recognize by this, or the receptor, number 2, inhibitory receptor. So when it binds here, and these activating receptors also there, and what will happen, it stops the activation of this to kill. So the MHC molecule bind to these inhibitory receptors, and it will command to the nucleus, and thereby the activation of the receptors or the cytokine putting the bomb, it will be stopped there, and thereby this normal cell escape from this phenomenon. So if you imagine this guy of the natural killer cell, as I mentioned before, and this is like a James Bond, like you have a license to kill. It has its own gun, and this have a 3 type of recepting capacity. One is been activating. This is the smoking gun, where it has a gun in his, and is a pistol where the all these bullets as a cytokine is a bullet to kill it. But when it recognizes the MHC1 molecule with the inhibiting receptors, as soon as it binds to the MHC1, the pulling the trigger is stopped. This is our own person, our own self cell. It's not a foreign cell. Don't kill. It is not an invaded one. When it lacks that, it will fire it, and then that cells will be killed. This is one part. Another part is the NK cell, the receptor antibody coated cell can bind with this receptor FcR3. This receptor binds with this antibody coated any type of antibody. Suppose it's a bacteria or virus, which is an antibody coated cell. It binds with that one. It can also kill that one, just like a antibody coated cells engulfed by phagocytosis when we studied the innate immunity system, and this is another way as an innate immunity. Do you remember that innate immunity? We have studied that one. So this is an both specific immunity and innate immunity put together, where the specific immunity help, or the antibody will help to activate the NK cell to destroy these infected cells. Follow that point? Any question? You can also, we'll check into your textbook in a figure. Yes, it is there in the figure 410. If you see that good picture of the cells, the inhibitory cells, probably you may know that, but I will go through it once again. Could you see this one here? Normal cell, okay? And here's the abnormal cells. And this cell is lacking a peptide. You look at the MHC class 1 molecule, and it is lacking here. So what will happen? These cells will activate the cytokine and kill this abnormal cell, which is lacking this. These cells will be destroyed, but not this cell, because this cell is having an MHC1 molecule here, and this molecule is being inhibiting this activation or to kill or pull the trigger. It stops that, and thereby it can survive the normal cell. Another type of antibody coated one, these are the antibody, the inverted Y shape is the antibody, and they are the antibody coated target cells. And these cells also being recognized by the NK cell, and it can trigger the suicidal of this particular death or kill this cell, antibody coated target cell. The function of NK cell, using 3 receptors, it can, one is activating and another one is recognition of the MHC1 and another one is the antibody. That's all. You have to write your short notes, okay, for NK cells. Now we shall go on to the next one. Okay. MHC molecule. What is this MHC molecule? We are talking about this MHC, right? Okay. What is MHC? We always talk about what MHC, major histocompatibility complex. What is MHC molecule? We call it as a major histocompatibility. So you have to write, if I ask about what is MHC, I'll give you a major histocompatibility complex or minor or histo and C for cat. Like, if I give different choices, you should remember like a major histocompatibility. Right, okay. MHC molecule, major histocompatibility complex, C for complex. Okay. So what I'll give you another 5 to 10 minutes a break, and then we will come back and we will go through this MHC molecule. Any questions? Any questions? No? Kelly, are you okay? Okay. Do you follow? Oh, okay. Okay, now we will go on to the details of MHC molecule. Okay, that's on the major histocompatibility molecule. MHC, to just I mention here, MHC molecule. And what is the MHC molecules? Initially they thought they are used to, they consider initially rejection of grafts. They are responsible for that, okay? And when this occur rejection, the exchange between different members of a species. Okay, example. We are all human beings. We belong to the same species. Okay. One person here, the human, okay, one can donate kidney to human 2. Same species. One human being donating a kidney to another being. But that is not being acceptable, and this human 2 started rejecting this one. This human want transplant kidney. Transplant. So this rejection from the exchange of organ from one person to the other, and this we call it as a graft, we call it as a transplantation. Transplantation surgery, right? Kidney, liver, okay? Transplantation of kidney, liver, or heart. So, if you organ transplant from one human to the other, the same, you are not talking about the transplantation of a pig heart to the human heart, or no, human to human. Even then, that is a rejection which is occur. Rejection of graft. the main reason for this rejection lies the MHC molecule. It's an MHC molecule. This is responsible for this rejection of 1 gram. So what do you understand from this? What do you think of this rejection? Why this rejection? Rejection is due to the MHC molecule, I said. So the MHC molecule is responsible for the rejection of the grafts, meaning human 1 will have one type of MHC molecule, human 2 you have a different type of MHC molecule. Okay, going to the mechanism, just I want to explain further. This is the one, okay, this is another species. You have the MHC molecule 1 and this is the MHC molecule, MHC class 1. This is a type of MHC also 1, okay. This is the human 1 and then human 2, okay. So could you see the structure of this MHC molecule? This is also a class 1 molecule, MHC 1 molecule, and this is also MHC 1 molecule. This is also, but this one is different from this. So when you get this part of the tissue, see all the other part, suppose if you have of a human, like a sticky diagram or this, okay, so this person will receive this, I put it like a heart like this, so remove and then put this one is here. So the shape of the MHC molecule or the molecule for this or the human 2 is a different shape than this MHC1 here. So what happens here at this junction? Suppose you are mixing this particular heart from human 1 is donating and you are transplanting the surgery here. What will happen here is the human 1, okay, you have the MHC molecule 1 and the another part of the tissue where you are donating the heart or the heart, this is the heart tissue. It will have like a MHC, a different type of MHC. So this is the persons and their NK cell can easily identify this MHC molecule and thereby it will recognize as a self, hey, I should not touch this. But when you put another person's heart and then transplant here, and this NK cell, these natural killer cells, and these cells will not recognize this. So what will happen if it is not recognizing that MHC1? There you go. This NK cell will attack the heart muscles at the junction, and thereby this heart is being rejected out, or kidney or any other parts rejected out, because this NK cell will eat these cells, attack these cells, and they say, hey, you are not belonging to me, you are foreign, you are not belonging to mine. I need this MHC molecule like this, and you looks differently. I am not recognizing you, get out. Started eating. Yes, go ahead.
Is that like where the blood types come in?
Correct. It's coming out, but that is a bit different one. But same mechanism is there. We are going to the blood type the next class also. Okay, we'll go through that. But this one we are talking about the NK cell, natural killer cells, and thereby it will reject. So this is the graft rejection and transplantation immunology, which we will study probably on the chapter later part of the course. We will study in detail. But just I want to inform you what is MHC molecule. The MHC molecules are the one which will function as a, in case of the transplantation rejection reactions. Okay? Okay?
Dr.
Yes.
Soma,
Go ahead.
I know you don't like writing in marker, but you really can't see when you write in pen.
Oh, okay. No, I'm all right. I can switch over to the marker. You are not able to see that one, the figure which I draw now? No?
No, sir. It's
Okay. I put it that way. This is a H1, and you have a heart. This is a heart. Okay. And this molecule is different, and this MHC molecule is in one way. Okay. So this NK cell which can recognize this and identify as a self. So it will not attack this cell, this particular cell, this human one. But this heart is coming from human 2. Another human being donated the heart, and we are transplanting to this person because this heart is not good. So what will happen at the time, this NK cell it can recognize this as self, it won't touch this. At the same time, this will attack this because it will not recognize this MHC molecule, and thereby it attack, and once it attack is established, most of the junction of the of the recipient as well as the host, I mean this is a host and this is the donating heart, it will attack and then it will go away, or the rejection part. So there is no use in using a transplantation. So we have to do some sort of immunocompromising thing. So immunotolerance, and then how to to keep this heart or this liver or the kidney intact with this. So those part is transplantation immunology, which we will study later part of the class, of the course, and another week. But now I want to explain you how the MHC molecule plays a major role in rejection of the graft. Do you follow that? Okay. One simple question. The eye transplant, which will be any rejection there? Eye? Question.
If you transplant the eye, will there be
Yes.
rejection? Could be, I guess, yes.
Rejection, entire eye, but eye lens sometime.
No.
No. Why? Not getting into the blood things over there. That's the question. Think about it. Answer me next class. Okay. What is the nature of these class molecules? Nature of these molecules. These MHC molecules. Okay. So there are two types of molecule which we have studied. Class 1, okay, and class 2. Okay. And both of them, class 1 molecule, class 2 molecule, both of them are membrane. Are membrane bound heterodimer. Now I want to explain what is this heterodimer. Anyone? Do you know about what is heterodimer?
Two
Two
strands,
protein molecule, yes.
two different protein
Molecules. Okay. So dimer means two, and hetero means different. So you can have it like a simple explanation. Two different proteins or polypeptide chain. Polypeptide chain. Two different. Okay. So class 1 is also having two different protein polypeptide chain. Class 2 is also made up of two different protein or polypeptide chain. We call it as a heterodimer. So you should understand what is heterodimer. What is homodimer means now.
two of the same
Yeah,
polypeptide.
same polypeptide, two polypeptide together, that is called dimer, but it's called homodimer. In this case, we call it as a heterodimer, where two different polypeptide chain, and these two chain are placed in the membrane, or cellular membrane. That's why we checked without these MHC molecules are present in the membranes, correct? And then to identify the normal and abnormal cells. So now we will go into the little bit how these peptides are arranged. Okay. It consists of, like I put class 1 molecule. I will focus it in a little bit, out of focusing now. I think probably this will happen. Okay. Class 1. MHC. Class 1 means here I am just a MHC class 1. Okay. Class 1, they consist of consists of, okay, heavy polypeptide of how much? 44 kilodalton. This is the molecular weight, KDA. If I say this is the molecular weight of this polypeptide chain, and they are non-covalently, this heavy polypeptide chain, non-covalently. You know what is covalent bond? You know covalent bond? Anything about chemistry? Do you know about the chemistry? How this one atom to the another atom, you know, they bind, or one element is bind to another element. The bond is ionic bond, covalent bond, and coordinative covalent bond, you know, all those details, right? So here is a non-covalently linked, okay, they linked, non-covalently linked to a smaller peptide. So I told you two peptide, how these two peptide is, one is the heavy polypeptide, another to a smaller peptide, they smaller 12 kilodalton polypeptide. And this 12 kilodalton polypeptide, otherwise we call it as a beta microglobulin. Globulin. Beta microglobulin. So one is the heavy polypeptide, another one is the smaller polypeptide, and they are non-covalently linked. So that is the class 1 molecule. You understand that? Class 1 molecule. So this class 1 molecule is the one which is a characteristic of a particular person. Meaning this class 1 of a person is unique. Just like, as you mentioned before, like a blood group. But here you can say, yes, it is a blood group type of thing. So the gene which coding for H1 or this class 1 for you is different for me. For me, and then different from her, and then you different from another person. It varies. And it also has a genetics on that part, MHC molecule. We will see the heavy polypeptide chain and non-covalent link to the smaller polypeptide chain, how they are organized in a very good fashion in your textbook. We will see that one too. Okay. The largest part of the heavy chain, the large part, okay, of heavy chain protrude outside, protrude from cell surface. So as I mentioned before, this is a cell, and it is coming out, right? It's coming out. It's coming out. So some part which is into cytoplasm and some part is outside, and this large part of the heavy chain which is protruding outside, that gives a signal, a green flag. Hey, I am all right. Everything is okay inside the cell. I can say, if you see, this is a small country, a small town, and then you see outside the surface of these are the territory, what I can call is at the edge or the surface surroundings of this city, is everything is normal. So green flag is okay. When the green flag is not hoisted there, what will happen? If the MHC molecule is not there, meaning this cell is already infected or it is a malignant cell. So there is a very intelligent way of communicating to the NK cell. So that's why the polypeptide chain which is protruding outside is coming the large part of heavy chain of MHC1 molecule. Okay? And there is another part, a hydrophobic section. Okay, another part of the same molecule, hydrophobic. So hydro means water, phobic means hate. So water hating section of this molecule, hydrophobic section. anchors in the membrane. Okay, and the short hydrophilic sequence is it present in the cytoplasm. Something like if I draw a picture, a cartoon, and this molecule, this is the membrane, it's a lipid biomembrane. Okay, so this lipid biomembrane is coming up like this. Now this molecule of the heavy chain, which is going out here, and it's coming here, this molecule, and now this is the hydrophobic region here, and then some of them are coming on to the C-terminal on the cytoplasm. This is cytoplasm, and this is the outside the surface, and this is the membrane bound. This is the membrane bound, this hydrophobic section, which is going to be on this membrane bound here. And this is the one, the large part, which we explained, and this is the cytoplasm. So this is the organization of the MHC molecule, okay? And we will see the next molecule is the class 2 molecule, MHC class 2 molecule. And this is also a transmembrane.
Dr. Silva, can you go back to the last slide, please?
Okay, this one? Okay.
Yes, sir. Just
So this is this hydrophobic section of this molecule. This is a one polypeptide chain, right? There and also it's the two, like a heterodimer. I can also draw the another one like a green color, and this is also there, like two different molecules. Okay, put together. I'm just like a cartoon, and I want to explain this part as a membrane, and then the cytoplasm part, and this is the membrane outside. So this part is really protruding outside as a surface, and this is the membrane part, is hydrophobic region, and this cytosol part. Okay. Done?
Yes, sir. Thank you.
Okay, good. So the next part is your class 2 molecules, and this is also a transmembrane, just like a class 1 transmembrane molecule. Otherwise, transmembrane glycoprotein, I put it, glycoprotein. Transmembrane glycoprotein. They consist of alpha and beta polypeptide. As I mentioned before, this is also the same thing. Like this molecule is alpha, which will have a 34 kilodalton molecular weight. Beta is 29 kilodalton, DA. This is a molecular weight unit, KDA, kilodalton, KDA, kilodalton. Okay. And they also have a considerable a sequence similarity with the class 1. They have similarity in the sequence with class 1 molecule, but the gene codes for this class 2 is different. Okay, we call it as the genes code for class 2. They call it as the molecule as HLADR. Okay, and the main regulatory sequence which control the transcription. So the class 2 molecule, which is also a transmembrane, and alpha and beta polypeptide, they made up of two protein, as I mentioned before, like heterodimer, and one part is 34 kilodalton, another one is 29 kilodalton. They code from genes. Okay, that genes we call it a HLA-DR. You just remember the HLA-DR. We will see more what is the HLA-ABC. That's for class 1 and class 2 is HLA-DR in a little bit. Okay. And then next part is the MHC3. MHC3, another type of molecule, but they have a different variety of other genes apart from class 1 and 2. They congregate with the MHC chromosomes. These molecules congregate. Congregation, like a congregate with the MHC chromosomes, and they grouped to form MHC3, class 3 molecule. The example of this is the C4 protein isotypes, we'll see later, and C2, and these are complements, 4, okay, and complement 2, I think we have studied in the earlier class, and factor B, these are the protein, and the tumor necrosis factor TNF alpha, and lymphotoxin, LT alpha and LT beta, LT alpha and LT beta. Okay, these are the molecule which is present, these genes are present in the same way of the MHC chromosomes. They congregate along with this, and thereby, but this function is different from this, but C4 and C2, they form as a complement factors, and the TNF alpha is a cytokine molecule, tumor necrosis factor alpha, and that will activate most of the cell suicide or the apoptosis signals. And the lymphotoxin is another one which is also involved on the cellular toxicity. So these forms as a MHC class 3 molecules. So I want to study MHC class 1 and MHC 2 and MHC 3, which we discussed bit earlier now. So now we will go on to the gene map of MHC. Yeah, so far any question? Any question? Okay, good. So we are going to find out some genes so that we can understand easily the mechanism of the gene map of MHC. Okay, MHC molecule. As I mentioned before, MHC molecule is different from person to person, just like your blood group substance. As I said, blood group. So when you mix the blood, a person is A group blood, and if you give him B group for the transfusion, what will happen? That's it. That's the end of it, right? So you cannot mix match. Yeah, this at the same time, if you mix with the A group with another A group, they can survive, right? Because they can match the blood group substance. The same way, if you have this MHC molecule of the same type, of the HLA type of a person, and then you donate the same tissue type, then they can match. That's why they used to do a tissue typing laboratory. Have you ever heard of the tissue typing laboratory? When you have the transplantation of a kidney or any other body parts to another person, the donor, and they used to check whether they are compatibility. Histocompatibility, they call it. Histo means tissue, and tissue compatibility in this sense, they have to find out this MHC molecule, or they call it the HLA type, tissue typing. And their results will depend upon this MHC presence or the similar type of thing, a donor as well as the recipient. So that part we will see that. So the gene map will tell you all about. So in the figure, in your other textbook 4.1.2, up and down regulation of HLA type, okay, and figure 4.13, 14, 15, they have a complete sequence of human MHC being available. So I will just go through that now, and then we will see the next part in your, okay, these are the class 1 molecule, how they are organized in the cell. MHC molecule 1 and 2. I'll focus 1 first and then I'll go on 2. Could you see that one here as a heterodimer? The beta 2 microglobulin as a small molecule, small peptide. This is a large one. And this is the one with the COH group, and this is the cytoplasmic end, and this is a membrane bound. And this molecule alpha helix and cleft, I mean, how the molecule is folded in the class 1. This is a class 2 molecule where you will find and this alpha 1 and beta 1 and alpha 2 and beta 2. And these are the molecule with the membrane bound, and this is the class 2 molecule. Both are the similar structure. Could you see the alpha 1, alpha 2 and the alpha 1, but here the beta 1, and then alpha 2 is here. It's not unlike the class 1. So it looks as a similar one, but the function is a bit different with the class 1 as well as the class 2 molecule. So the next one is the figure 412. You can see the gene map of this one. The TNF interferon gamma, and they are activating this particular gene sequence to synthesize more of MHC class 2 molecule, HLA-DR. It will synthesize. So these are the gene sequence and how they activate the Tata box and the leader sequence, and these are the protein which is being transcribed into the protein. Okay, but the LPS, otherwise the lipid polysaccharide, which is mainly coming from bacteria, and the interferon here the alpha and beta, and they downregulate. Okay, that's in the macrophage of the same HLA2 is being inhibited by this lipopolysaccharides, which is from the macrophage as well as from the bacterial infection, or even the interferon alpha or interferon beta, they downregulate here to synthesize. So the gene for the HLA DR alpha, that is depend upon the availability of LPS as well as the TNF mechanism or TNF regulation. Okay, and that another one is for 13 here. You will see the details of the HLA gene and the gene products. We can see the HLA-B and HLA-C, HLA-E, HLA-A, HLA-G, HLA-F. These are the gene products. These are the human one side, another one is for the mouse. So they study the MHC class 1 gene map. You will find HLA-B, HLA-C, and HLA-A. So these are the gene sequence. And they in the mouse, you have K, D, L, and Q, T, and M. So if you go on the MHC class 1, you have a HLA-B, C, A, E. Okay, and then the same thing in the HLA-2. Sorry, in the MHC2, you get the DP and DO, DM, and DQ. So there are different varieties of the protein, and they have got different proteasome complex peptide transporters, and the same thing mechanism which they proved in the animal model with the proteasome complex and peptide transporters. I'm not going to the details of individual protein expression or the gene expression, but I want you to understand that HLA, DP or DO or DQ or DR, those are the products of MHC2 class. HLA1 you will find, HLA-A, B, and then C, they are the product for MHC class 1 molecule. So that's what you want to remember, that one. Okay. And the composite difference in the MHC molecule, I'll just come in. They call it as the haplotype. Haplotypes. What is haplotypes? As I mentioned before, MHC class 1 will have a HLA-A, HLA-B, and HLA-C, and HLA-G. It goes on, HLA-G. Different type. Come on. Okay. These are, they call it as a haplotype for the MHC class 1 molecule. Okay. And the class 2, I'll go on to the next one. Class 2, what is haplotype? Haplotype. Just like a blood group, we call it, right? In this case, we are talking about haplotype. The same thing, but in a different way that we are expressing it. Here we have a HLA-DR, HLA-DO, HLA-DQ. Okay? So these are the varieties. HLA-DP. Okay? So they are the sequence. These are a particular gene is expressed. This gene is expressed. This is another gene is expressed. So individually, all of them are class 2 molecule, but the type is expressed vary. Do you understand that? The same thing is here. MSC class 1 molecule, but the type of A, B, C, G. It may vary. So we are all human beings. But one is called like a George, another one is a High, and another person. The same thing is here. MHC class is a one group of that particular molecule. But individual person, you may express HLA-A or B or C or G. So these are the same gene, but it variant. How do you call it that type of same gene, but it may vary person to person, that gene. That variation, that variability, we call it as, we call it one name, alleles. Do you know about it? Type of genes? Have you ever heard of that one? Alleles, right? So that alleles, we are talking about this one, haplotypes. Yep. Do you follow? Little bit of genetics, not too much, a little bit, otherwise Dr. G will explain to that one detail. I'm not going to go on those details, but please remember that HLA, HLA-B, C, G. Okay. Like blood group substance, individually you have your own HLA. Do you know about it? Your own HLA type? No, I know mine, because when I did my graduate studies, I have a tissue type card. Like here, you can call it as a tissue typing laboratory. Do you see that one? Tissue typing lab, Department of Immunology, School of Biological Sciences, Madurai Kamaraj University, Madurai, India. When I did my graduation study, and the immunology department, and they tissue type everybody there to check it out. Okay? So when you do that one, and then we have to have the Professor R.M. Pichappan, but he's now retired, but this is on 9/12/83. Okay, at the time when I was graduating. So the result is here. This is my own tissue typing. HLA. Could you say HLA? Report? What is that? What I have now? HLA 2, the HLA AA 1, and then HLA B 1340. So this number represent on how much I have this HLA here. Okay, predominant. And then here the HLA B I have a 13 and 40 here. I'm just focusing here. Now, okay. And my blood group is O and Rh positive, and this is haplotype, and these are my detail. These are the haplotype. Okay. If I have this card, if I need any of the transplant of any body parts of my own, I need not to worry about it. I can put it on a database. I want to check it out. Hey, who have this type of things? Immediately we can get those guys. Hey, can you donate your kidney for me? I will give you thousands of dollars and millions of dollars to you. So if you have this, and you can bargain with that one. I mean, that's the part which the medical community, they will handle, because there's a privacy and everything on that part. Okay. So this is a one simple example, and this is in future. This we have done, but so many typing now is more advanced and more levels, and I haven't tested my recently what is my tissue type. It will never change, but just in case, I want to check that out. Okay, so it's still possible. In our lab, we want to screen all of you, your HLA type, and then keep your card like this. Okay, so that is a neat part. So I want to know whenever if you are being asked, hey, you did immunology, you know about the tissue rejection, you know about histotype or tissue type, and you can very able to say, hey, I know that, you know, a tissue typing laboratory or any other HLA type, whatever HLA type you have, and you can ask doctor, what is HLA type? Even doctor will explain to you, hey, you know what is HLA and what is MHC and everything. I want to know, this is my type. And so that way, you know, make sure you got a proper kidney or transplantation, otherwise you can sue the doctor. Okay. Any question? Now we'll go on to the next part. The different MHC haplotype, which I mentioned before, and they comprises of, the haplotype comprises of 224 genes, loci. Okay, 224 gene loci that involve. Okay, and 100 out of this 120 genes. Okay, that codes for 40% of immune system. Okay, immune system function, I put it in that way. 224 gene loci, out of which 120, nearly half of the genes, code for 40% of immune system function. So the largest part, okay, that polymorphism, the largest part of this polymorphism. What is polymorphism? Based upon the multiple alleles. You know, morphism, you know, morphology looks like a genetics polymorphism. Have you ever heard of that one too? No? The variety, just now I mentioned before, you have the HLA class 2 molecule, they have HLA DR, DO, and all. So all these, what is that? They call it haplotype, and the gene level, we call it, they have the alleles. So this is one gene, this is another gene, the same loci, but the arrangement of the gene is a bit different. So that's why the person who is a short, one person, another one is a tall, another one is a skin color is different, a dark, another one is a fair, another a brown color eye, or gray color eye, or hair color. So all those things, we have the gene for hair, gene for skin color, but that gene is different from that one. So that difference, we call it as alleles. Do you follow that? So the same way, the alleles of the class 2 is the HLA, DO, DR, DO, DQ, different type of alleles. Gene is class 2, MHC class 2 gene, but they have different alleles. Can I explain that one too? Yeah? Do you follow? Okay. So now this allele, we call it as not one allele, not two alleles, we got is it? Multiple alleles. So they express, you know, from one person is different from another. So they morphology, the different protein, we call it as a polymorphism. Why there's a polymorphism? Because you have multiple alleles. Multiple alleles. They constitute for the polymorphism. Okay. Now, go on to the class 1 and 2 genes are the most polymorphic genes. polymorphic genes in the entire human genome. So that's very important. We know now the human genome. In the whole human genome, the most polymorphic genes are the class 1 and class 2 genes. Do you follow that? Meaning you have multiple alleles and multiple genetic morphism, polymorphism. So most polymorphic genes that they are from human genome is class 1 and class 2. That you should understand that. So you get, how much you can get it now? 200 allelic variants. Allelic variants. Okay, they've been identified so far. 200 allelic variant identified, and they have got, as I mentioned before, HLA2, HLA-DROQP, and another one in the on the class 1 we did with the HLA-A, B, C, G. Okay, they are polymorphic. They are polymorphic, and they class 2 also polymorphic. Okay, the next one is the MHC region loci. MHC, I put it the same thing, just I'm explaining different way. MHC region is a outstanding hotspot. Hotspot, okay? They are the hotspot with mutation. Hotspot with the mutation. And this mutation, how the mutation they form for this hotspot? They form different types of mutation. A, recombination mutation, recombination, which you know already in genetics, recombination, and you have a point mutation, yes. So they constitute the hotspot for the hypervariable region, just for the different types of MHC molecule. Also, they have a homologous unequal crossover mutation. Then the D, you have a gene conversion. Okay. So MHC region is the one more variable region, and that variable region is due to the mutation, and that forms a different type of polymorphism, and the type of mutation may be a recombination, type of mutation may be a point mutation, and the type of mutation is an unequal crossover between the genes. Another one is the gene conversion. So all these constitute for the hotspot. Okay. Any questions so far? No? Okay. Just for the next one is the inheritance of MHC. How this MHC is inherited. Inheritance, MHC molecule. Okay. In the animal model, they have studied. Whenever we study about the inheritance, you know about the F1 generation, F2 generation, like. So if you have a pure strain of mice, pure strain, meaning they made brothers and sisters of these mice, okay? Brothers made sisters, and they produce a homozygous for each pair. That meaning homologous chromosomes. So they have a pure strain. Okay. When they cross-react, then it follows like a Mendelian inheritance, like a haplotype of these MHC molecules. They have the haplotype of the inherited molecules, like a mother and father is going to be the same one. That is the pure strain. When you cross this pure strain, like a H2K versus plus H2D, this is a pure strain. This is a pure strain, and this is also a pure strain. When they do, what you get, H2K1 and H2D. Another one is the H2KD, right? You have, this is a hybrid variety. And when you breed this hybrid, again H2KD and KD, you will get the K and KD and D. So the first generation, you get this only, right? When you get that one. When you breed this, you get the pure strain, another pure strain, another a hybrid, KD. And this is a part of an animal model experiment, and they proved to be for the MHC molecule. That is true. That is a part. Okay. This is the simple genetics on what we did study on. The tissue distribution of MHC molecules. Just one more thing, like a tissue distribution of MHC molecule. Tissue distribution. As I mentioned before, all nucleated cells, they have class 1 molecules. All nucleated cells have class 1. That's why the NK cell cannot touch the nucleated cells, and this is a normal cell, it will go out. But all the other cells which is infected, this the expression of the class 1 is not, then that cell is be killed by NK cells. And also these class 1 molecule, they expressed in mostly, abundantly in lymphoid tissue. Lymphoid and myeloid cells. And it is less, this is the most abundant. Then less in liver, lung, kidney. And then very rarely or sparsely present in brain and skeletal muscle. So, as if in all nucleated cell, but there is a category. Mostly your lymphoid cell and myeloid cell, and less in liver cell and lung and kidney, and then sparingly or very rarely you find the brain and skeletal muscles. These are the class 1 molecules. Okay. And the next one is the surface. This one more thing is the human surface. Another, the same MHC molecule, surface of in the human being, surface of placenta, extra villus cytotrophoblast. This very interesting one is here. This human surface, the placenta, you find the class 1 molecules. They call it as a HLA. They don't have that one. The placenta, they lack, they don't have this HLA-A and HLA-B type. They don't have it. Though they have the nucleated cell, but they don't express. They call it as a allodeterminants. The HLA-A, HLA-B is not present in, is a lack, it's not present, they are not present in placenta. So what do you suggest from this? Placenta, they don't have HLA-A or HLA-B, they are the allodeterminant. But they may express an HLA-C, but that is not like an allodeterminant here. So this is the alternative type we call the HLA-G, and that is also not as a allodeterminant. That meaning in the tissue rejection, the HLA-A and B are involved. So that's why when I showed my card, you will find here HLA-A and HLA-B, not HLA-C or D, no. So HLA-A and B, they are allodeterminant, and they don't express in placenta. So whenever you have a tissue transplant, For example, in one incident example I know, a person who is suffering from a chronic ulcer in his leg. Any medication, any antibiotic, nothing. He is having chronic ulcer in his leg. So the doctor, he took a placenta for anyone of this, after the delivery. He took it and then he did a transplantation onto the ulcer. He got cured and no more ulcer from his leg. So if any other tissue, it is not. But the placenta, they have stem cells, and the placenta, they have enough rich components of other cellular ingredient of the growth factors, and that helps in healing. At the same time, that tissue of placenta is never rejected out because they don't have HLA-A or HLA-B, allodeterminant. So that's one point just I want to make sure. HLA lacking part is in placenta. Okay. Now the class 2 molecule, now let's go on the class 2 molecule. They are highly restricted in expression. They present in B cells. They are present in B cells, dendritic cells, B cells, dendritic cells, D-E-N-D-R-I-T-I-C, dendritic cell, macrophage, and thymic epithelium. Thymic epithelium. When it's activated by gamma interferons, you get the endothelial cells and epithelial tissues that can express this. So class 2 is expressed in B cells and dendritic cells and macrophage and thymus epithelium. This is a normal one, normal expression. Okay. But when you treat with a gamma interferon, another cytokine, interferon, I-N-T-E-R-F-E-R-O-N, interferons, when you treat, then you get an expression of the class 2 in capillary endothelial cell. Endothelial cells and epithelial cell. If you know in histology what I am talking about, you may understand. Epithelial cells and tissues, and also they expressed increased levels of class 1 as well because of the gamma interferon that induces class 1 as well as class 2 in this increased expression. So the main function of these MHC molecules, MHC function. Now we'll go on to the function, main function of MHC molecules. MHC molecule function as a surface marker, as I mentioned before, surface markers, yes, enable the infected cells, right? Surface markers, the infected cells don't express and thereby they get killed, and cytotoxic helper T cells. They are expressed in those cells, and rich in polymorphism. They express in polymorphism. And why you have this MHC molecule? Give protection. Correct? Because it gives and identify the cells, and thereby it can protect our system from the diverse microorganisms. One of the example here for the protection is from the malaria. If you have a malarial infection, okay, Plasmodium falciparum, Plasmodium falciparum, and they express in East Africa strain of Plasmodium. They associated with HLA DRB1001, okay, 10101. This is a start. This is the one type of HLA. So it activates, is associated with this HLA, Plasmodium falciparum of East Africa. And the same thing for the West Africa, a West African strain of the Plasmodium, and that will converse with the HLA DRB1302, and this is the gene which is being activated, and East African gene activated this, West African gene activated the another one of this, and they confer for the resistance. Resistance of what? Resistant to malaria, malarial infection. Okay, if this malarial parasite infected, getting into our system, getting into our cells, if it is from the East Africa, you get HLA, this which is being expressed because of this HLA type in your system will protect from Plasmodium. So it will activate your immune response, and thereby it will kill this one. Plasmodium infection will be suppressed. If you have a West African falciparum which is being infected, then this type HLA which is present in your system, class 2 molecule, that will help to protect. In either way, class 2 molecules plays a major role in protecting yourself from any other infection. Okay. The last one is the non-classical MHC molecule. That's on the MHC3 which we did study. Non-classical MHC. And these molecules, like a MHC-like molecule, not MHC molecule, the MHC-like molecules. And they are called CD1, and they present the lipid and glycolipids to T cells. They also help, CD1. And HLA-E, this is another type which is also non-classical MHC molecule, and they signals peptides from classical 1 to CD94 and NK cell G2 receptors. So they are not like a MHC molecule, but the MHC-like molecule, they perform the function of MHC molecule. They present antigen presenting cells. So that is the part which we did study onto the chapter 4, and with this which I am finishing that, and the next one is the chapter 5. Perhaps we can go on to the next class. I would like you to go through the chapter 5 before coming to the class, and all this animation a little bit, and thereby you can you can understand the concept at least. Just I give the one context.
One
Probably that will
monoclonal
help.
antibody recognizes an
Do you see now
epitope
from
on sperm whale
Synchrorange
myoglobin,
and
which is dependent upon amino acids
Victoria?
at positions 34, 53,
Okay.
and 113 in the primary amino acid sequence.
I don't know whether you have the
Only when the molecule is correctly folded do these three
Sound
amino
system
acids
is
come to be
working.
in close proximity to each other, so
Could
that
you
they are
listen
able
or
to
could you
interact
hear
with the antigen combining
any
site of the
sound system?
monoclonal antibody.
No?
This type of epitope
think
is
here the
referred
sound
to
system is
as
not
a
working.
discontinuous
I would
epitope.
like you
That
to
is,
watch
the amino acids
this
are far
in
apart in
your
the primary structure,
in
but
your
become close together
leisure
once the
time
molecule is
onto
folded.
this all this animation from your
The
website,
majority of
and
antibodies
thereby
appear to recognize
you will get more
discontinuous epitopes.
understanding, and then we will
Thus,
go on to the next
a
class
second monoclonal
about the
antibody raised against
interaction,
sperm
the
whale
primary
myoglobin
interaction
recognizes
with
a
the
structure
antigen.
that is
So
critically
now we
dependent
know
upon amino acids at
about
positions 83,
the
144, and
so far we know
145.
about these receptors, which is the
Again,
receptors we are
this is a
talking
clear
about, and the
illustration of a
MHC is one type of
discontinuous
receptors
epitope.
and different type of receptors we did study.
However,
Now the antigen
some
is coming,
antibodies do
so this
recognize
interaction,
what is
how
referred
this
to as
antigen
a
speaks to
linear
these
or
receptors.
continuous epitope.
Now what will happen?
A third
when they-
monoclonal antibody raised against
these
sperm whale
receptors, and
myoglobin
what will happen
recognizes
next
a
step?
structure
So
that is
those
dependent upon
details we
amino
will
acids
see
at
in the
positions
next class.
18,
Okay.
19,
So I
20,
want you to
21,
have you
and
seen
22
the
in the molecule.
any videos on your
This
website?
is clearly an example
Writing your
of a
summary.
linear epitope.
So if you have attended the
Approximately
class and
80 to
collecting the
90% of
notes, class
antibodies
notes, and
probably
writing the summary
recognize
of these
discontinuous
class notes
epitopes,
and post onto the
whilst perhaps 10
WebCT
to
email, that is
20%
your
of
homework.
antibodies
Okay,
recognize
I haven't done it for the
continuous
last
epitopes.
week. Some of you submitted,
perhaps
but
important
I
to
will. I
appreciate
look
that an
forward.
epitope
I
can
will
be
grade them
partially
up.
linear
That
in
will be
nature,
added onto
with
your
additional
final grade.
discontinuous
So I'm
amino
just
acids contributing
repeatedly I'm
towards the
saying
epitope.
to you, every class when I finish, that
Binding
week
to
is
the
the
linear
due. For the
sequence,
next
which
week is the
forms
due
part
for
of the
that. So
epitope,
if it's a
will be
backlog,
of a
you
lower
please
affinity than
do it
binding
from
to
the
the
last
complete
lecture onwards.
epitope.
I'm not worrying about the previous one. From the
The
last
amino
lecture
acid at
onwards,
position
write a
109
summary
has been
and submit
identified
onto
as
the
being
WebCT
critical
email.
in a T
That
cell
is your
epitope of sperm whale
assignment, your
myoglobin.
homework for grading. purpose.
It will be recalled
Is that
that
right?
T cells
Any question
recognize
from
linear
Synchorant?
peptides derived from